Okay, well, I think we can make a start anyway. So, good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. Now, with this series, we hope to shake up the usual format of lecture series by having a mixed format with time dedicated to the presentation, demonstration, and discussion of software toolkits or methods. Typically, these sessions will run for about 50 minutes in total, and we'll run the series throughout the year on the last Thursday of each month. Now, you can already see that we've got a great lineup, and we have for the next few months on this slide shown here, and we're really looking forward to all the upcoming talks. If you are interested in presenting a new method or software you have been developing or implementing, please feel free to reach out. So before moving on, just a quick few introductions. My name is Lewis Jones, and with me today I have Erin Dillon and William Gertie. The three of us are on the development team for Palaeoverse, an organization aiming to bring the paleobiology community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about the Palaeoverse, please check out our GitHub organization, and we'll just drop those links into the chat now. And you can also check out our website. But that's all from me for now. I will now give over to you, Will, who will give some helpful tips on instructions for this webinar, followed by Erin, who will introduce this month's speaker. So thank you very much, and please enjoy today's lecture. Over to you, Will.
Thanks, Lewis. As Lewis mentioned, the format of the seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end dedicated to question and answer and discussion. My fellow hosts and I will help field your questions and moderate the discussion. You can use the, oh, sorry, I got ahead of myself. Please note this seminar will be recorded. The recording's already begun. This recording will be made available on the Palaeoverse YouTube channel. And we will stop the recording before the question and answer period in case you prefer not to be recorded when you ask your questions. Just quickly point out a couple of useful Zoom controls. At the bottom of your screen, you should see a Q&A button. You can use this to submit questions to the speaker at any time over the course of the talk. During the Q&A session at the end, we'll then read out your question or invite you to turn on your microphone if you'd like to ask the speaker directly. You can also use the raise hand button. And again, during the question and answer section, we will then invite you to turn on your microphone so you can ask your question. The whole time you can chat with the hosts in case you have any technical difficulties, but we'll also open up the chat to allow you to chat with the other attendees during the question and answer period. There's also a closed captions button that you can turn on to view the transcript of the talk at any time. Lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. With this in mind, we ask that you please be respectful and kind when engaged with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, you can refer to our Palaeoverse Code of Conduct, which can be found on our website, and I believe the link is being put in chat now. And with that, I'll pass it over to Erin to introduce today's speaker.
Wonderful. Thanks so much, Lewis and Will, for bringing us all up to speed in terms of how the lecture series is going to work. So today I have the great honor of introducing the second speaker in our lecture series, Dr. Benjamin Shipley. So Dr. Shipley is a postdoctoral research fellow at the University of Oxford and a member of Dr. Aaron Saup's lab. He obtained a PhD in biology in 2023 from the Georgia Institute of Technology in the US, where he worked with Dr. Jenny McGuire. Dr. Shipley's research explores the relationships between species ranges, global patterns of diversity, and the climatic and geographic factors that influence how species are distributed across space and time. He is particularly interested in how small-ranged and endemic species evolve, survive, and react to environmental and landscape changes. To approach these questions, Dr. Shipley integrates a variety of GIS modeling and machine learning methods. And ultimately, by understanding the drivers and the dynamics of species distributions in both the present and past, his research aims to inform the conservation of vulnerable species. So today we're thrilled to hear Dr. Shipley present a talk entitled Modeling Species Range Shifts in the Past and Future. So please join me in welcoming Dr. Benjamin Shipley to our virtual Palaeoverse stage and take it from here.
Awesome. Thank you so much, and thank you all for tuning in. I am honored to be invited to give this talk about modeling species ranges. The work I'm doing today, that I'm going to talk about today, is work that I did in collaboration with several researchers at Georgia Tech, where I got my PhD. So during this talk, I would like to highlight some of the challenges that we might have when modeling species distributions in the past, and new advances in methods that can help mitigate some of these challenges. I'll talk about the software package I developed, Mega SDM, and I'll apply these new methods to a case study of the Florida mouse, looking at its range shifts through the Pleistocene. To note, all of the code and the software that I will use in this discussion, in my demonstration, is on GitHub, and it's also published in a journal article in Ecography. So it should come as no surprise to anyone here that Earth's climate has changed in the past, and it will continue to do so in the future. And these changes are non-uniform through time. Looking at this graph here, we see global temperature change on Earth from the last 66 million years. And there are periods of warmth, like the early Eocene, and there are periods of cool, like the Pliocene and Pleistocene. So it's clear that it's not uniformly increasing or decreasing in temperature. In addition, these climate changes aren't uniform across space. Looking to the future now, we can see that anthropogenic climate change in the next century is going to have different effects in different locations across Earth. For example, the Amazon rainforest is predicted to get hotter by about 7 or even 8 degrees Celsius by the end of the century, whereas areas in Southeast Asia might only increase by 3 or 4 degrees Celsius. This relationship is even more piecemeal when we're looking at precipitation, with areas that are predicted to get drier, like Southern Europe, and areas that are predicted to get wetter, like the Southeast Pacific Islands. So the complex spatial dynamics of climate change on Earth lead to similarly complex dynamics in the biosphere, and in particular, how species might shift their ranges in response to climate change. So this map here shows a hypothesis as to where species might go as our climate continues to warm in the United States. We see amphibians in yellow, mammals in pink, and birds in blue. And although you can see the general trend towards the north in the areas of warmer climates, we do see a lot of variation in where species might be going to flee from maybe more inhospitable areas. However, this map is somewhat simplified in that it shows where species should go if we're able to track climate fully, but many species are not able to. So it's much more complex than even this map might suggest. And therefore, understanding how species shift their ranges in response to climate change is a fundamental question in biology today. In particular, looking to the past, we can provide additional fundamental insights into the ecology and conservation of certain species. For example, a paper published in 2016 looked at the historical range changes of the desert horned lizard, which is in the southwestern United States, finding that it may have originated in the Sonoran Desert and expanded its range as climate warmed. However, the researchers also found that the species is more likely to be more generalist than expected. It's able to live in wetter and cooler areas than it does now. In addition, In addition, we see that path range shifts can tell us a little bit about the conservation of certain mammals. We know that historical range contractions can predict extinction risk in mammals, even when those species have re-expanded their ranges again. So knowing that path range shifts are important, how might we evaluate these? Well, one of the main ways that scientists do this is by using species distribution models. So broadly, species distribution models are hypotheses about where species live based on certain environmental variables. They link observations of a species to environmental suitability across the landscape. And I'm just going to give a short little crash course into species distribution modeling just to make sure we're all on the same page before I talk about why it might be a little bit difficult to apply species distribution modeling to geographic range shifts in the past. So in general, species distribution models compare occurrence points and absence points across the landscape. So you need occurrence points where you observe an animal, or in the case, or a plant, or in the case of paleontology, maybe a fossil record. And then you also have absence points where the species is not found. Although true absence points are not very easy to get because it's hard to know where a species is not. A lot of models nowadays don't necessarily require real absences, but instead pseudo absences, simply sampling the available climates in your study region. And then these models also incorporate environmental layers, which are going to govern the species' response to the climate and what habitats are necessarily suitable for them. So these can be continuous climate variables like climate or precipitation. They can be other environmental variables like topography or distance to a river. They can also be categorical like soil type. And then finally, these models use an algorithm to relate the environmental conditions of occurrence points to those of absence points. Finding areas that might be more suitable for the species. And ultimately, they create these habitat suitability maps. This is one I created for the pygmy rabbit using MegaSDM. So the areas in green are areas that are highly suitable for the pygmy rabbit, and it's generally speaking in the Great Basin and the Columbia Basin north of it. And then the areas in red and gray are areas that are less suitable for this species because it's got different environmental conditions. So despite their ubiquity in the ecological literature, modeling range shifting species is often very difficult. Most commonly used SDM methods are overly simplistic when applied to range shifts across time, assuming that climate change changes uniformly either across space or across time, which we know it doesn't. So as a result, I and several of my colleagues at Georgia Tech developed MegaSDM, which is a modeling tool that attempts to kind of bridge the gap between using habitat suitability models and looking at how that actually influences where the species change along the landscape. This modeling tool is specifically developed to evaluate range shifts in the past and present, and it uses the Maxent software, a machine learning algorithm for the actual modeling. But you can input other models if you have given them as well. So although MegaSDM does a few different things to improve our estimates of where species live on the planet, including environmental subsampling, which attempts to reduce bias in our occurrences and absence points, and also new background sampling methods. But I'm going to focus on two aspects of MegaSDM that are particularly relevant for paleontology. Its incorporation of dispersal rate and also multiple time steps. So let's start with dispersal rate. Broadly speaking, dispersal rate is the ability of the species to move across the landscape. If a species is a good disperser, it can move across the landscape very easily. If it's a poor disperser, it might not be able to. And in fact, we know that dispersal rates limit the ability of species to track climate across the landscape. A paper in 2012 found this in mammals in the Western Hemisphere. The dots below this line here, let me turn on the pointer. The dots below this line here are species that have dispersal velocities less than the velocity of current anthropogenic climate change. Therefore, these species are unlikely to be able to track climate and are going to have to contract their ranges as the warmest areas of the ranges become inhospitable. And this is actually appears to be the case for many species across mammalia, but this is especially bad in primates and shrews. So to take an example of how this might influence species distribution modeling, let's do a conceptual example. Let's say a species lives in this green polygon in the year 2000. I can model the suitable habitat for the species in the year 2050. Let's say the species is a warm adapted species. So as our climate warms, there's more suitable habitat for the species. But if the species is not able to disperse throughout all of the habitat, it's not really suitable for it. Maybe it can only disperse to an area in this smaller polygon around the original range. That makes all of the other suitable habitat unreachable, which leads to a dichotomy between modeling the species niche, which attempts to look at the suitable habitat, versus the species range, which tends to look at the habitat that the actual geographic area that it will be able to reach. This is a really big problem in paleontology, as given that that species past distributions influence their current ranges as well. And in fact, this has been found to influence species distribution models in a variety of papers. One of them, published in 2020, found that looked at tree modeled species, used species distribution models on different trees, tree species throughout the Pleistocene in North America, and found that the predicted ranges using the models were much larger than the observed ranges using fossil pollen. But nowhere is this more egregious than in models looking at the expansion of the nine-banded armadillo across the United States. So the nine-banded armadillo first arrived in the United States after bridges were put across the Rio Grande River in the late 1800s, and since then has been steadily moving north into the east, and now it lives pretty much everywhere in the southeastern United States. However, this species is a generalist species, so if you're modeling the distribution, the potential range of the species, as the authors here did in 1996, you might find that areas on the West Coast are suitable and provide suitable habitat for the species. However, it's nearly 30 years later, and records of the armadillo have not reached the Western United States. This is likely because the Rocky Mountains provide a complete barrier to dispersal for these species. So how does MegaSDM deal with dispersal constraints on species distribution models? It explicitly models these dispersal rates by modeling their dispersal as an exponential distribution given as a particular rate. So to use the Franklin's ground squirrel as an example, this is a small ground squirrel that lives in central United States. It has a pretty slow dispersal rate. It's not able to move very fast across the landscape, about 1 kilometer per year. So MegaSDM models takes that rate and models the probability that the Franklin's ground squirrel will be able to disperse any given distance, and then multiplies this dispersal probability by the habitat suitability map. So I've to illustrate how this works in practice, I've created 2 different maps here. The map on top shows the predicted distribution of the Franklin's ground squirrel in 2030 without dispersal constraints. And you can see areas in Alaska and central Canada have opened up as being suitable for the Franklin's ground squirrel. However, the Franklin's ground squirrel doesn't live in Alaska and Canada at the moment, as well, the areas of Canada that it is. So it might not be able to colonize these areas. And instead, we have to incorporate this dispersal rate to show that this species is not going to reach Alaska. So now that we've talked about dispersal rate, it's time to move on to looking at multiple time steps and how that might provide nuance into species range shifts. So I mentioned before that climate does not always change unidirectionally through time. And in fact, this graph here shows this pretty clearly. This is a graph of temperature from 21,000 years ago to 10,000 years ago. And we see lots of small fluctuations in the temperature, and we even see a few longer protracted periods of cooling within this general period of warming. So these transient climate changes can influence species distributions today. For example, I'll give you a bit of another conceptual example of how this might work. So let's say there's a species that lives in this green polygon in 1800. And then following some sort of environmental shift, it shifts its range to the west in 1900. Now, if the climate shifts back to the east, there might be areas that are suitable for the species opening up on the opposite side of its original range, but because it's already shifted its range to the west, it might not be able to reach them. So Mega SDM deals with these transient climate changes by using a multi-step analysis approach. So most SDM most species distribution model software today only use one or maybe two time steps, training your model on one time step and projecting it into the future or to the past. But Mega SDM, you can put an arbitrary number of these time steps in and model them each sequentially. And ultimately, what this ends up outputting is what we call time maps, which show the piecewise expansion and contraction of species across the landscape. So this animation here shows, again, using the Franklin's ground squirrel and dispersal constraint from 2010 to 2070. And you can see areas in red are those that the Franklin's ground squirrel has vacated, and areas in blue are those that they've expanded their ranges into. But then even more interesting are the areas in purple, which might be hard to see, but I'll put my pointer right next to them. These are areas where the species has expanded their range from time step 1 to 2, but then contracted it again. So it's these transient changes that end up providing more nuance into how species actually shift their ranges. So now I've talked about two very briefly aspects that might improve our understanding of how species shift their ranges. I'm going to apply them to a case study of the Florida mouse. So the Florida mouse, Potamys floridanus, is a species of mouse that is endemic to Florida, but in particular to the temperate grasslands of central Florida. It's near threatened globally, but it's also just rare. It's not very common even within its species range, and it's got a very slow dispersal rate of only about 0.03 kilometers per year, which makes it kind of an interesting case study to look at here. And I'll be modeling range shifts in the Florida mouse from the last glacial maximum 21,000 years ago to today using 5 different time steps. I've pulled the climate layers for these time steps from the Palaeoclim dataset, which was published in 2018. And I thought it would be interesting to look at how this species was able to shift their ranges throughout the Pleistocene. So now I'm going to give a short walkthrough of Mega SDM. Again, to note, all of the code and all of the files that I'm going to be using to make this will be available on GitHub. I will put the link in the chat after this walkthrough. So the only thing that Mega SDM requires to start is a set of climate data. So I'll show you what I have already. I've downloaded the climate data from Palaeoclim, and I've got all five time periods, starting with the current one and going to the last glacial maximum 21,000 years ago. And each of these folders has the same environmental layers in it. These are all bioclimatic variables. So Bio 1, for example, indicates mean annual temperature, whereas Bio 12 is mean annual precipitation. So I have these climate data, and I can start, and that's the only thing that Mega SDM requires to start. So I pulled up the demonstration code here. First, I've got to load the package itself, and I'm also going to load a package to plot to give you some examples of what might it look like, some of the intermediate steps might look like. So before starting, we have to set our working directories. I'm going to set it to that desktop folder you just saw. And then we're going to define an output for all of the data and the models that we're going to create in Mega SDM. So that's going to be, I'm going to call it the Palaeoverse folder. And then the first thing we have to do, which is the same thing for every species distribution modeler, is you first have to load and manage your climate rasters. Ideally, all of your rasters, these are continuous image data showing continuous variables, for instance. And these are data that ideally would be in the same projection, same resolution, and the same extent across the different time periods and the different variables. However, I know in practice, this is not the case. So the first function that Mega SDM has standardizes all of that, both for the training area, which is the current data that we're going to train the model on, and the data in the future or the past. So to do this, we're going to just take a list of the environmental files. We're going to make a list of them, and I'll print this out here in the console so you can see. This is just a list of the raster files we have. And then we'll do the same thing for all of the historical and paleoclimate layers. So now I've got the same type of list for all of the different 5 time periods I'm using. Then we can use this first function from MegaSDM, train study env, to standardize all of these climate layers and make sure they all have the same resolution, they have the same projection, and they've got the same extent. And you can change the extent of your area using this function as well. If you have global climate rasters, but you don't want to model your species globally, you can do this here. So I'm just going to plot one of the climate rasters so we can see what it looks like. And you can see that it's most of the southeastern United States. Palaeoverse floridanus is endemic to an area in central Florida, so there's no need to go with a larger extent than this. So now that we've got our training climate rasters managed, it's time to do the rasters in the future or the past. And again, for this example, I'm using 4 different time periods in the past. So first I have to get those into a list, but then I can use the predict env function here, highlighted in MegaSDM, to do the same type of management, to get them in the same format as the training data. So I'll just run this. One thing you have to note about this function is that you do have to write out when your time periods are. And this is especially useful if you're going to incorporate dispersal rate, as dispersal rate is generally speaking distance per unit time. So if you have, you'll need definite times to incorporate dispersal rate. So we've got all of our climate data now. Now it's time to look at the to manage our occurrence data. Although most people in that will be species distribution modeling will probably come with their own occurrence data. MegaSDM does provide the option to download occurrences from GBIF, the Global Biodiversity Information Facility, if you so desire. So that's what I'm going to do here, just to give you an example of what it looks like. And this will take a second, but in the meantime, this is essentially just a wrapper for the RGBIF function, although it also cleans the data in a different way to ensure that we're only getting the highest quality data from GBIF to be used for the model. So when this finishes, I'll move on to the occurrence management section, which will help us to manage our occurrence data in a way that makes it more relevant to use for the model. So I'll show you what the occurrence data looks like at the moment. We can see that I've created a new folder here that says occurrences. And if I click the Palaeoverse floridanus CSV file, we get this large spreadsheet, which is essentially all the data that GBIF provides when you download it from, when you run a query from GBIF. And so we have a bunch of stuff that is very useful for citations and for for error checking, but maybe not so much useful for species distribution modeling itself. Really, the only thing that we care about are the coordinates and the name of the species. So one of the things that the occurrence management function does is it removes some of those columns and then extracts the environmental data, which are the things that we're interested in anyway. So now the same file has only the species, the coordinates, and the extracted climate data from each of the points. So now that we've collected our occurrence points, we can create our background points. As I mentioned before, true absence data is very hard to come by and also very costly to create. So most species distribution models, including Maxent, which is the algorithm that we're using, requires pseudo absence points, which simply attempt to sample the available climate of the background and relating it to where we know the species does exist. Although the best method of generating background points is still up for debate. Every species distribution modeler has their own preferred method. I'm going to do a buffer constrained background point method where we sample a lot of background points within buffers around the occurrence points. This will help to not to make sure that our models aren't artificially being inflated in terms of how well they might predict given species distribution. And also it helps to make sure that we're not getting biased by we're not by sampling issues and spatial bias there. So I've created these buffers now. I made, you can set how wide you want your buffers to be. This is in meters, so I've set the buffers to be 75 kilometers around each occurrence point. And I will plot that now. You can see that the buffers are around where the species lives in Florida. So now that we have the buffers, we can sample the actual background points. Again, just everyone has their own preferred method of sampling background points, but I'm just going to use a very basic way to do it, which is to sample 1,000 background points. But one of the interesting advances that MegaSDM does in terms of background point generation is it allows for you to sample a proportion of background points within buffers versus outside of the buffers. So if I set the spatial weight parameter to 0.5, half of these points, 500 of them will be taken from within this buffer, and then half of them will be taken from outside. And this kind of serves a dual purpose. Again, it stops the model from being overfit, but it also stops over extrapolation in areas that we don't have the species. And then we can decide whether we want to environmentally sample these background points or not. We did environmentally sample the occurrence points, so we should probably do the same for the background points. Then I'm just collecting a list of the buffers. There's only one buffer in this case, which is the one I made just now. And then we're going to just sample the background points and create our set of 1,000 or so background points. To note, when you're the way that the environmental subsampling works, you won't necessarily get exactly 1,000 background points because there might, depending on how clustered the data are, you might end up removing more or fewer points. So now I'm just going to show you what the background points look like in the file Explorer. Whoops. Here we go. Backgrounds. And as you can tell, it's formatted in the same way as the occurrence points, with the species, the coordinates, and the climate layers that have been extracted from the data. So now we can finally move on. We've got our environmental variables extracted from both our occurrence and our background points. Now we can actually use modeling. And to run the model, MegaSDM uses the actual JavaScript for Maxent. I know there's an R package specifically for it, but the JavaScript provides a bit more fine tuning than the R package itself. So we decided to use the JavaScript directly as opposed to the R package there. And you can see it is done now. I've run 4 replicates of this in order to come up with more of a consensus model of where a species might live and how it might be influenced by different environmental variables. And I can show you the results of this in general, and they're going to be in the models folder here. And we can see a bunch of different files. In particular, the Maxent results file provides validation metrics, so like so AUC values, true skill statistic, validation metrics like that. And then there are HTML files that provide all of the outputs that Maxent provides natively in its JavaScript form. So now that we've done that, we can project, we've created these models in environmental space, but now we need to project it into geographic space, both in the present time and also back through time. So to do this, we'll use the Maxent Proj function in MegaSTM. But first, we have to define what our time periods are and our different climate scenarios. MaxEnt allows you to run multiple different climate scenarios at once. So you could use different, if you're projecting into the future, you could do different representative concentration pathways. Or if you're looking in the past, you can do different paleoclimate models. We're going to define the directory where the study area, our training area rasters are found, and then define a list of all of the folders where our projected climate layers are found. And then we can run the MaxEnt approach step, which will take those environmental response curves of the species to each of the environmental layers and project them into space. This is going to take a second, so I've actually already run these to provide an output for you. And so I'll just show that while this is running. No, not this. So the output of the MaxEnt approach step is a set of rasters that provide habitat suitability maps ensembled for the 4 different replicates, so taking the median of each of the replicates, and also binary presence-absence maps that are created from thresholding this habitat suitability. They give MaxEnt generally, MegaSTM generally gives raster data for it, but it also prints out PDFs of the different ranges. So we can look at those right now. I'll open these up. These are the ones, I'll open up the ones for current time at the moment. So we can see binary. The binary map here shows binary presence absence in the Florida mouse in the year 2000. And then we got the same thing, but this is just habitat suitability. And then if we look at it back through time, we can look at the different, the hypothesized distributions of these species back in 21,000 years ago. So as you can tell, it's quite different. The climate in Florida was somewhat different in the last glacial maximum than it was today. And we see that instead of being found in Florida, as in central Florida, as you might expect, these species in 21,000 years ago may have been found more preferentially in areas like the Bahamas and southern Florida. So after running the MaxEnt approach step, we can create those time maps that I was mentioning, looking at the piecewise expansion and contraction of species ranges. And to do this, we simply take the binary maps of each of the species, each of the presence-absence maps for the species in each time period, and overlay them, looking at where the species lived at each given time. And again, because this is going to take a little bit of time, I have the results already up. And we'll go back to this. So the time maps I created yesterday for the Florida mouse show pretty significant changes in the species range from 21,000 years ago to today. I'll walk you through a little bit about how I would interpret these maps. First, we see a lot of red in the south, which indicates that the species is contracting the range, which means that it may have originated in the south and expanded northwards. As the climate warmed and Florida became more hospitable for the species, it may have dispersed northwards, colonizing central Florida. But we see that there's this large pink area here around Florida, and this is where the species expanded their range as the climate became suitable, but then contracted it as ocean levels rose and the area was inundated. So from this map, it seems these models suggest that the Florida mouse actually did not natively originate in central Florida, but instead had to disperse there from the south. So that's it for this demonstration, but I just wanted to leave on a bit of a soapbox and say that you have to know where you've been to know where you're going in terms of modeling species distributions. Species distributions on the planet today are a function of not only their ecology now, but also historical contingencies. And as a result, just using dispersal rate and multiple time steps do add lots of nuance into these species distribution models. And but it is important to know that species distribution models in general and mega SDM in particular should be used to build hypotheses about how species shift their ranges, not test these hypotheses, and that they should always be validated by the paleontological record and other methods to see how well it might hold up. Again, the code I've used to make these demonstrations can be found on my GitHub. I will send a link in the chat, and I'm happy to answer any questions. I will continue running the code I've started while people have questions. Thank you so much.
Thanks so much for an excellent talk. So let me go ahead and stop the recording.